Let's have an image-based discussion in Helminthology. Helminthology is the study of helminths. This is the first image showing eggs of Enterobius vermicularis. Enterobius vermicularis is a nematode. So the egg of Enterobius vermicularis has a characteristic plano-convex shape. So D-shaped egg, plano-convex shape. These eggs are non-bile stained and they float on saturated salt solution. Coming to the next image, this image shows the egg of hookworm. There are two types of hookworms. First is Ankylostoma duodenale and the next is Necator americanus. The eggs of Ankylostoma duodenale and Necator americanus are similar and they cannot be distinguished. This is the outer membrane and this is the inner membrane. The outer membrane of the egg is regular and smooth, whereas the inner membrane is irregular. It resembles the structure of a mitochondria, and the embryo is arranged in the form of many blastomeres. The hookworm egg is also non-bile stained and floats in saturated salt solution. This image shows the fertilized and unfertilized eggs of Ascaris Lumbricoidus. The Ascaris Lumbricoidus is also a nematode, commonly called as roundworm. So the Ascaris produces two types of eggs, fertilized eggs and unfertilized eggs. The fertilized eggs are round in shape. These are the fertilized eggs which are round in shape and it has an embryo inside. This is the embryo and there is an albuminous coat surrounding the outer membrane. And on either sides of the embryo there is a chitin sheath present, this and this. At the two poles there is presence of chitin sheath surrounding the embryo. This is the fertilized egg of Ascaris Lumbricoidus and this fertilized egg floats in saturated salt solution. This is the unfertilized egg of Ascaris Lumbricoidus. It has an irregular outer membrane and an atrophied embryo within it. This unfertilized egg sinks in saturated salt solution. Next picture. This image shows the egg of Treacherous Trichura. It has a characteristic barrel shape. This is a barrel shaped egg with two mucus plugs at the poles which protrude out. So these are the mucus plugs at the poles of the egg which protrude out, giving it a barrel shaped. Next image. This is the egg of Schistosoma Hematobium. The egg of Schistosoma Hematobium has two membranes, outer membrane and the inner membrane, and it has a characteristic terminal spine. Based on the arrangement of the spine, the three species of schistosoma are differentiated. Next image. This is an image showing the egg of Tinea. Both Tinea solium and the Tinea saginata. Pork tapeworm and the beef tapeworm. The egg of Tinea shows a striated, so these are all the striations present. Striated inner membrane which is thick and brown in color, and the embryo contains 3 pairs of hooklets. Three pairs of hooklets. So this is the egg of Tinea, which has a thick inner membrane with the presence of brown and thick striations. The embryo has 3 pairs of hooklets. These are the hooklets. We have already compared the fertilized and unfertilized eggs of Ascaris Lumbricoidus. This is an image showing only the fertilized egg of Ascaris Lumbricoidus. It has a thick outer shell and a proteinous coat. And there is an embryo within it. The embryo on either side of the pole is covered by a chitin sheath. Next image, this is an egg of Schistosoma mansoni. This is another species of Schistosoma. So the same in Schistosoma Hematobium. It has an outer membrane and an inner membrane, and there is characteristic lateral spine present. Schistosoma Hematobium had a terminal spine, whereas the Schistosoma mansoni has a lateral spine. Next, this image shows the egg of Schistosoma Japonicum. Same as in other two species, it has an outer membrane and an inner membrane, and the presence of characteristic lateral knob. There is characteristic lateral knob present in Schistosoma Japonicum. This is a comparison between the three eggs of Schistosoma species. The Schistosoma Hematobium has the characteristic terminal spine. Schistosoma Japonicum has a characteristic lateral knob, and the Schistosoma mansoni has the characteristic lateral spine. Next image, this is an image of Fasciola busci, and this is an image of Fasciola hepatica. Both these are trematodes, that is flukes. So the Fasciola busci has a tapering end. Whereas the Fasciola hepatica has a protrusion. This is the way to differentiate between the Fasciola busci from the Fasciola hepatica. This is an image of Tinea solium, that is the poke tapeworm. The parts of Tinea solium, it has a head, that is calyx, and then the neck, followed by the strobila, which has many proglottids. The structure of the head, it has the presence of rostellum with 2 rows of hooklets. So these are the 2 rows of hooklets. And it has 4 suckers present. So the differentiation between Tinea solium and Tinea saginata is based on the presence of this rostellum and the hooklets. The rostellum with the presence of 2 rows of hooklets are seen only in Tinea solium, and they are absent in Tinea saginata. rostrum and the 2 rows of hooklets. And these are the images of the hookworms. Ankylostoma duodenale and Necator americanus. These are differentiated by the presence of the mouth parts. The Ankylostoma duodenale has teeth-like projections which are 6 in number, whereas the Necator americanus has plates which are 4 in number. They cannot be differentiated by their eggs, as both of the eggs are same. The Ankylostoma duodenale causes more iron deficiency anemia compared to Necator americanus. This is because the Ankylostoma duodenale has a sharp teeth-like apertures which causes more blood loss and thereby leading to more severe iron deficiency anemia. This is an image showing hydatid cyst disease. Most commonly affected organ is the right lobe of liver. The hydatid cyst disease is caused by Echinococcus granulosus, which is the dog tapeworm. This is an image showing the egg of Hymenopus nana. H. nana. Hymenopus nana is a cestode. Coming to the characteristics of the egg, it has an outer membrane and an inner membrane, and there is a space between the outer and the inner membrane which contains yolk granules. So all these are the yolk granules. And there is an embryo situated in the middle. Containing 3 pairs of hooklets. And the characteristic feature in this Hymenopus nana egg is there are polar filaments running between 2 poles in between the outer and the inner membrane. So here they can be visualized, polar filaments. This is the egg of Hymenopus nana. As you have seen earlier, this is the unfertilized egg of Ascaris Lumbricoidus. This unfertilized egg has an irregular outer membrane It has a smooth inner membrane and an atrophied embryo present within it. This is an image of hydatid cyst. The hydatid cyst is the larva of the dog tapeworm Echinococcus granulosus. Coming to the morphology, it is made up of 3 layers: pericyst, ectocyst and the endocyst. The pericyst is derived from the host membrane, whereas the ectocyst and the endocysts are derived by its own. Next, the endocyst is responsible for the production of hydatid fluid. Which is responsible for the causing of disease. Next, it is also in continuation with the Brood's capsules. These are the Brood's capsules. So the endocyst is in continuation with the formation of brood capsules containing the daughter scolex. This is about the hydatid cyst morphology. This is the image showing the female and male worms of Ascaris Lumbricoidus. That is the roundworm. The female worm is larger than the male worm, and the end of the male worm is curved. But the end of the female worm is straight. This is also a picture of hydatid cyst disease. Hydatid cyst seen inside the body. This image shows the scolex, that is the head of Tinea solium. Here the rostellum along with the 2 rows of hooklets can be clearly visualized. Rostellum with 2 rows of hooklets. So this is an image of Ankylostoma duodenale, which is a hookworm. This is the egg, in which the outer membrane is smooth, the inner membrane is irregular, and it has the embryo in the form of blastomeres. This is the rhabditiform larva, and this is the adult worm. This image shows a condition called as cutaneous larva migrans. Caused by the migrating larva, most commonly seen with Ankylostoma. The larva of Ankylostoma causes cutaneous larva migrans, in which there is creeping eruption seen most commonly in the extremities. As we have seen above, this again shows an image of the male and the female roundworms. That is Ascaris Lumbricoidus. The longer one is the female worm, which has a straight pointed tail, and the smaller one is the male worm, which has a curved end. These are all the important images in Helminthology. Thank you.